Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we need to talk a little bit more about how cells get things into a cell and out of a cell and through that membrane. We've talked about the phospholipid bilayer before and all of its special properties, so let's take a closer look. So just as a review, this is a cross-section of a cell membrane, the phospholipid bilayer. And we said, hey, this thing's got some really unique properties because those circular regions of the membrane, those are hydrophilic heads. And all those tails that are in the center are hydrophilic tails. And so we have an amphipathic phospholipid bilayer, so it's really difficult to move things into and out of the cell. It is selectively permeable. Now you can see some proteins here, and they're going to play a role in helping things get into and out of a cell as well. So let's take a closer look. Let's see how that actually happens. All right, so now we have to define some terms. This picture here is showing us three terms, and I want you to imagine that that black line is a cell membrane. We have the environment on one side and inside the cell on the other. The first one that we're going to deal with is actually in the center. Isotonic solution. In an isotonic solution, what we're saying is those black dots, which represent solute, could be salt, could be anything, but solute is going to be the same on either side of the membrane. So whether it's the environment or inside the cell, the concentration of solute is the same. And so the water movement across the membrane is going to be the same. The amount of water leaving the cell is going to be the same as the amount of water entering the cell. And so that's an isotonic solution. On the left-hand side, we have hypotonic. Hypo means low. Now, in this particular situation, what we have in a hypotonic solution, on the left-hand side there, that shaded area is going to be outside of the cell. Inside the cell is going to be where you have more of those black dots, and that's going to represent the cytoplasm. In a hypotonic solution, water is going to go from where it is more concentrated on the left-hand side across the membrane to the right-hand side on that hypotonic part of the table. The goal here is to balance out the concentration of the solutes and the water on either side of the membrane. In a hypertonic solution on the far right-hand side, what you see is the environment outside of the cell has more solute, more black dots than there are in the cytoplasm. And therefore, what's going to happen is water is going to go through the membrane, leave the cell. Now, what we're talking about here is the process of osmosis, where water is moving from where it is more concentrated to where it is less concentrated to reach equilibrium on either side of a membrane. All right, so now let's take a look and see what's going to happen to a cell when placed in each one of these environments. When a cell is placed in a hypotonic solution, there is less solute outside the cell relative to inside the cell. Therefore, water is going to come from the environment, move into the cell, and you're going to see the result is cell lysis. In an isotonic solution, again, we said that the concentration of solute is equal compared to the environment and inside the cell. And so water is going to move into and out of the cell, but it's going to be at equilibrium. On the right-hand side, we have a hypertonic solution. So we have more solute outside the cell than inside. Water is going to leave the cell, and the cell is going to shrivel. All right, so in a plant cell or a bacterial cell, cells that have cell walls, the outcome is going to be a little bit different. In a hypertonic solution, what's happening? Again, the solute concentration is higher outside the cell relative to inside the cell. And so water is still going to leave the cell, but because there is a cell wall there, when you look at it under a microscope, you're going to see that the cell has maintained generally the same shape, but the cell membrane is pulled away from the cell wall, and we call that plasmolysis. Isotonic solutions, there really isn't going to be much of a change. In a hypotonic solution, water is going to be entering the cell. And as that happens, that cell membrane is going to push up against the cell wall, and we call that turgor pressure. So besides water moving into and out of a cell, cells do have mechanisms to bring in solute. Now, there's three basic ways that can happen. One is called simple diffusion, and in simple diffusion, you have solutes that just cross through a membrane. In simple diffusion, usually that's going to be very, very small uncharged particles or gas, things like CO2 and oxygen, that can move through a cell membrane. If a particle is too large to get through the membrane or it's charged, sometimes we have something called facilitated diffusion that allows things to travel from where they are highly concentrated to where they are low. And so in both simple diffusion and in facilitated diffusion, we're going from high concentration to low. The difference is in facilitated diffusion, facilitated means assisted, that process is being assisted by a protein that you see here. And so we're going from high concentration to low, but the solute is going through a protein. And then the next one is called active transport. And in active transport, what's being transported is going from low concentration to high. And because of that, you need a protein, but you're also going to need an input of energy to get that to go, you get the solute to go against the concentration gradient. Now, cells have mechanisms for moving larger particles into and out of a cell as well. Endocytosis would be an example of being able to take large particles and move them into the cell. And there's basically three primary ways. Penocytosis, which has been described as cell drinking, where the cell basically surrounds extracellular fluid, and whatever's in that fluid is going to be brought into the cell via a vesicle or a vacuole. Phagocytosis is when a cell is actually bringing in large organic compounds. And so one of the important examples there would be when you have a macrophage or dendritic cell in the immune system that's surrounding a bacterial cell or viral particles. That is usually done through phagocytosis. And then you have receptor-mediated endocytosis, which is a very specific type of endocytosis where there is a receptor for a specific type of compound. And once that receptor is triggered, it will allow the cell to bring that in. And cells will also get rid of large organic compounds or maybe secretory proteins through a process called exocytosis, where you have a membrane-bound vesicle that will fuse with the membrane and release the substance outside of the cell. All right, so that is a brief overview on how cells move things into and out of a cell. As always, thank you so much for being here. Have a great week.